Hello again, folks. This is David Villar, and welcome to this fifth video on toxicants with mixed effects on the CNS. On this video, we will discuss thiaminase containing plants and polioencephalomalacia in cattle. So there are two major plants that are toxic to ruminants and horses, which are bracken fern and horsetail. But thiamine deficiency can also take place in cats when they're fed fish diets that contain thiaminase, and occasionally in dogs fed cooked meats, because cooking destroys thiamine. It has also been described in fur-bearing animals like mink and foxes, and marine mammals like sea lions, seals, bottlenose dolphins, where it's called chastic paralysis. Now, starting with bracken fern, this is a perennial herb that is found all over the globe, especially in forest areas, because it likes the shade. And it can also be found in abandoned fields, open woods, and there are many reports of intoxications in cattle and horses. Now, there are several toxic principles that are recognized. Thiaminase is the main one for horses, which do not produce thiamine in their GI tract. But for cattle, it is tachylicide, which is a carcinogen that causes aplastic anemia and hematuria from tumors in the urinary bladder. On the right figure, we see what happens in horses. The thiamine gets destroyed by thiaminase before it gets absorbed. And without thiamine, there are two dependent reactions that become compromised. One is the conversion of pyruvate to acetyl-CoA, and the other is the conversion of ketoglutarate to succinyl-CoA in the Krebs cycle. So the animal is going to suffer from lack of energy and no production of ATP. Now, as we saw in the video of things that predispose animals to eat toxic plants, in this case it is the lack of better forage, or when the hay becomes contaminated and is fed. Both the fresh and the dried plants are toxic, and the signs start showing after significant consumption, such as, for example, 1 month of eating hay containing 20% bracken. The clinical signs differ because between horses and cattle. In horses, they're mainly neurologic, but in cattle, they are mainly due to bone marrow damage. And as we see here, in cattle, the disease is called a radiomimetic disease because they produce thiamine in the rumen, and therefore they are resistant to the thiaminase syndrome that is seen in horses. Now, the signs will start after 1 month of consumption, more or less, and include fever, aplastic anemia, hematuria, and in fact, the syndrome is also known as bovine enzootic hematuria. And the animals will have not only anemia, but also leukopenia and thrombocytopenia, which makes them prone to bleeding problems, not only in the urine, but also in the feces and nasal discharges and other places. If they live long enough, they will develop tumors in the bladder and the esophagus. With regards to horses, the clinical course is neurological, but very prolonged, to the extent that they develop emaciation, they lose a lot of weight in spite of maintaining their appetite. They will be lethargic, ataxic, and that has also been called as bracken staggers. They will have a wide stance with an arched back, and if they're not treated right away, they die with terminal convulsions in a matter of a few days. Now, in these images, what we see is the squamous cell carcinomas in the upper digestive tract of a bovine, with some small papillomas. And in the distal esophagus, there is a large carcinoma as it enters into the reticulum. Now, the treatment in horses is to give thiamine right away, daily for 2 weeks. And in cattle, really, there is not much we can do. We can try with blood transfusions for the anemia, which can be given along with protamine sulfate, which is a heparin antagonist. They also recommend broad-spectrum antibiotics because of the leukopenia, making them prone to infections. And the other thiaminase-containing plant is horsetail. This is also a perennial herb that can reach up to 4 feet tall. It's very easy to identify because it has hollow stems which are segmented, and the leaves are vestigial in whorls at the nodes. Now you can find it also worldwide along meadows, ditches, ponds, and wet areas in low-lying areas of pastures and fields. The toxin is thiaminase because ruminants produce thiamine, so it's just a problem for horses, but not really in cattle or sheep. There are no reports in ruminants. The most common source is contaminated hay. The signs in horses are the same as bracken fern, basically a nervous disorder and muscular weakness after approximately 1 month of daily consumption. And as with bracken fern, there is weight loss with continuous ingestion, and it usually ends up with the animal dying with convulsions. The treatment is the same with thiamine, and the animals are reported to make a quick recovery if we catch them early enough. Now, in dogs and cats, it is reported when the animals are fed cooked diets, because cooking destroys thiamines, fish, raw fish for cats, and it can happen with chronic anemia or when the cat is on a strict diet of only fish. There are numerous reports that contain thiaminase. Well, actually, there are numerous fish, I should say, that contain thiaminase, whether they come from fresh or salt waters. In marine mammals, the condition is called chastic paralysis. In foxes and mink, which are bred in captivity and fed raw fish, it has resulted in outbreaks of a spastic paralysis disease, which usually happens 1 month after the introduction of the raw fish in the diet. And in all the cases reported, the disease stopped once the fish was removed from the diet. Before the neurological signs appear, the animals begin to refuse food, and this also may contribute to develop a deficiency of thiamine. In cats, in the early stages, they develop GI signs of anorexia, salivation, vomiting, and weight loss, and these are followed by vestibular ataxia. They circle, they have this metria, hypersensitivity, dilated pupils that are unresponsive to light, and eventually they develop generalized seizures. And these convulsions usually end up in coma and death. On this video, we have a cat that was fed BFF canned food, and he's showing vestibular ataxia. As you can see, he's circling and falling, kind of tipping forward on one side all the time. And this was an incident that of certain batches of BFF, also known as best feline friends, that was recalled back in 2017, and there were about 300 suspect cases, and they were all traced back to eating the same thing. And when they measured thiamine in those canned foods, it was not even detected. So this is what an animal with a thiamine deficiency would look like. Okay, let's move on. Thiamine deficiency results in polioencephalomalacia, which means a softening of the cerebral cortical gray matter. But now we are going to talk about polio in cattle, and the cause is not a thiamine deficiency, but a high intake of sulfur with the diet, the water, or both. In the rumen of cattle, sulfur gets converted to hydrogen sulfide, which is almost exclusively a problem in beef cattle, particularly in feedlots. On this graph, what we see is the correlation between the concentration of hydrogen sulfide in the rumen, shown as the red line, and the numbers of animals developing polio in the columns. And at the x-axis, what we have is the days since they arrive at the feedlot. Now we're going to talk about the toxic concentrations in a minute, and in this case it was the water that had a mean sulfate concentration of 2,300 parts per million. We can see that after 10 days of arriving at the feedlot, the concentrations of hydrogen sulfide started to peak in the rumen, and that correlated well with the number of animals showing clinical signs. Now the rations with byproducts of corn, like distillers grains, usually have high concentrations of sulfate, and in feedlot cattle is the most common source when they are transitioned to a new diet. Now, how is polio diagnosed? This is a pathological diagnosis when the animal dies. What can be found is a mushy looking appearance in the superficial cerebral cortex, and when we examine those brain sections with an ultraviolet light, they autofluoresce, and you can see those that yellow discoloration in the same areas as you have on the upper image. Now, as we can see on this graph, high sulfur intake results in excessive production of hydrogen sulfide because of the ruminal microbial reduction. That hydrogen sulfide gas accumulates, and with the eructation of the animal, the gas is inhaled and it reaches the brain, where it is going to interfere with the energy metabolism and cause brain damage. The mechanism of action appears to be the inhibition of cytochrome C, which is an enzyme of the electron transport chain that is involved in the production of adenosine triphosphate. There are studies actually that measure thiamine in blood of the animals with clinical signs of polio, and the levels are normal. So we can say it's not a thiamine deficiency issue. Now, NRC recommends less than 0.4% sulfur in the diet. For water, the level of sulfate should not exceed 500 parts per million, with 1,000 being the maximum tolerated. Now let's see how we can calculate the total intake of sulfur by cattle. They send us to the lab a batch of corn hay mix, corn gluten, and water. And the first thing is to calculate the moisture content of the feed, because we're going to interpret everything on a dry matter basis. Next, we need to estimate how much the animal is drinking and eating. Water consumption is going to depend on the water temperature. If the temperature is low, about 40 degrees, we can assume that a cow is going to ingest about 8% of its body weight. So for a 500 kilogram cow, this would be about 40 liters. Now, the dry matter intake is about 2.5% of the body weight. So for a 500 kilogram cow, this is about 12.5 kilograms. And we need to express everything on a percent basis to compare it with NRC recommendations. So here we have the calculations for every ingredient of the diet and the water also. If we start with the corn hay mix, the first thing is to convert sulfate measured in the lab to elemental sulfur. Sulfur is only 1/3 of the sulfate molecule. If the analysis detects 816 parts per million of sulfate, that will equal to 269 parts per million of sulfur. So if we convert from parts per million to percentage, that equals to 0.03% sulfur. Now, if the ration, 90% of the ration is corn hay, the total intake of sulfur will be 0.03%. And we do the same thing for corn gluten, following the same steps. And if 10% of the ration is corn gluten, the overall intake by gluten is 0.04%. For water, when we convert from sulfate to sulfur, there is 62 parts per million, and if the animal is drinking 40 liters of water a day, the total consumption by water is going to be 2,500 milligrams. We have to add this value as if the water intake came from the ration, and because the animal is eating 12.5 kilograms, the total amount that comes from water is 200 parts per million, which is the same as 0.02%. And finally, we add all three sources of sulfur, 0.03 plus 0.04 plus 0.02, and the total sulfur intake is 0.1%, which is okay and far below 0.4% recommended by NRC. Now let's look at another example doing the calculations a bit differently from the previous case. Here we have the value of sulfur already present in corn gluten and the total mixed ration after accounting for the weight of the sulfate molecule, and we have the sulfur in water. So in this case, we're going to calculate the total amount in milligrams that come from the diet and water, and then we'll divide by the total amount of intake in the diet. So what we do first is taking away the moisture content to convert into a dry matter basis. Once we do this, we end up with 0.19% sulfur on a dry matter basis in the total mixed ration. And if we multiply it by 8 kilograms that the animal consumes, the total number of milligrams with the diet will be 15,200 milligrams. Next, we have to add the amount of intake by the water, assuming that the animal drinks 36 liters a day, and that's going to be 18,000 milligrams. And finally, we add the value from the diet and the water, and the total intake is 33,200 milligrams. That when divided by 8 kilograms, it results in 0.4% sulfur on a dry matter basis. Now let's look at the final example. This is simple because they already estimated the sulfur in the total mixed ration after accounting for the moisture content. We have the value of sulfate in the water, and we are going to assume the animal weighs 500 kilograms and the water intake is about 10% of its body weight when the temperature is 70 degrees Fahrenheit. We're going to assume a total dry matter intake of 3% body weight. So the question is, how much sulfur is present on a dry matter basis? Now, if we start with the water, the first thing is converting from sulfate to sulfur, and once we do this, we multiply by 50 liters of water intake, and that turns out to be 41,500 milligrams, or what is the same for 45 grams. Next, we calculate how much comes from the total mixed ration, and if the animal eats 3% of its body weight, that turns out to be 15 kilograms. The concentration of sulfur in the TMR was 0.2%, which is 2,000 parts per million, or milligrams per kilogram. And when we multiply by the amount ingested daily, which is 15 kilograms, the animal is consuming 30,000 milligrams of sulfur, which is the same as 30 grams. So the total amount ingested from water and feed is 71.5 grams, and when we convert it to a dry matter basis by dividing by 15 kilograms, it turns out to be like 0.48% sulfur. Now, in this case, there is a very significant amount that is coming through the water, and remember the water intakes varies a lot depending on the on the weather temperature. So if we use a value of 8% water intake, that applies for a temperature of about 40 degrees. But if the temperature is 90 degrees and it's summertime, the intake can go up to 18% of the body weight. So the total intake of sulfur will be more than double. So in this case, the calculations were done with 50 liters of water per day. But in summertime, it can double that number, and if the animal was drinking, for example, 90 liters a day, the final result would go from 0.48 up to 0.7%, which is definitely toxic. Now, this is the final slide. On the next video, we will continue talking about more toxicants that affect the CNS. So having said this, take care, and bye-bye for now.